4 billion years ago, Earth was not alive. No oceans filled with organisms, no oxygen to breathe, no cells, no DNA. Only rock, fire, and violent impacts from space. And yet, somewhere in this chaos, chemistry crossed a boundary. At some moment we cannot directly observe, non-living matter became the first living system. This is the story of how that transformation may have happened. At this moment, 4.54 billion years ago, the Earth forms from the debris of a collapsing cloud of gas and dust. Rock fragments collide, heat builds, the surface becomes a global ocean of molten magma. This is the Hadean Earth, a world defined by instability. Over tens of millions of years, something critical begins to change. The surface cools. A solid crust forms, thin, fragile, and constantly reshaped by volcanic activity. Vapor released from the interior condenses. Rain falls, not for days or years, but for thousands of years. And slowly, the first oceans begin to form, around 4.4 billion years ago. But just as conditions begin to stabilize, another force intervenes. Between 4.0 and 3.8 billion years ago, the inner solar system enters a period known as the Late Heavy Bombardment. Asteroids and comets strike the young Earth relentlessly. Each impact releases energy equivalent to millions of nuclear explosions. Oceans may have boiled away, only to reform again. The atmosphere may have been stripped, then rebuilt through volcanic outgassing and cometary delivery. And yet, paradoxically, this violence may have contributed essential ingredients: water, carbon compounds, and reactive minerals. By the end of this era, Earth possesses something extraordinary: a stable surface, liquid water, and a chemically active environment. The stage is set.
Now we ask a more difficult question. When did life actually begin? The answer is not straightforward, because the earliest evidence is subtle and often contested. Some of the oldest clues come from 4.1 billion-year-old rocks in Western Australia, where scientists have detected carbon signatures that may indicate biological activity. But these signals are ambiguous. Non-biological processes can sometimes produce similar patterns. So the search continues. More widely accepted evidence appears later, around 3.7 billion years ago in Greenland. Here, layered rock structures, known as stromatolites, suggest the presence of microbial communities. These formations are not individual organisms, but the result of countless microorganisms trapping sediment over time. They represent ecosystems, not life in isolation, but life already interacting with its environment. And yet, even here, debate persists. Some scientists argue these structures could still form without biology. Others point to their organization and chemical composition as strong evidence of life. What we can say with confidence is this: by at least 3.7 billion years ago, Earth was no longer lifeless. It was inhabited by simple, microscopic organisms, most likely resembling modern bacteria. Life had begun. But this raises a deeper question. Did all life descend from a single origin?
Every organism on Earth, bacteria, plants, animals, shares a remarkable set of features. They use DNA to store information. They rely on RNA and proteins to express it. They follow the same genetic code. This uniformity points to a profound conclusion. All life likely descended from a single ancestral population. Scientists refer to this as the last universal common ancestor, or LUCA. LUCA was not the first life form. It was the last organism or population from which all modern life inherited its core biological systems. But even this idea is not universally agreed upon. Some researchers propose a more complex scenario. Instead of a single ancestor, early life may have existed as a network of precellular entities constantly exchanging genetic material. In this view, life did not begin as a single lineage, but as a dynamic system of evolving chemical populations, gradually stabilizing into true cells. Either way, one conclusion remains unavoidable. Life did not appear fully formed. It emerged through stages, bridging chemistry and biology. At the heart of this story lies one of the most profound questions in science. How did non-living molecules begin to replicate, evolve, and sustain themselves? This transition is known as abiogenesis, and while no single theory fully explains it, several competing models attempt to describe the process. Imagine a molecule capable of both storing information and copying itself. This is the idea behind the RNA world. Unlike DNA, RNA can act as both a genetic blueprint and a catalyst. In laboratory experiments, scientists have demonstrated that certain RNA molecules, called ribozymes, can promote chemical reactions, including their own replication. This suggests a possible early stage of life where RNA existed before DNA and proteins, a world of self-replicating molecules, competing, mutating, evolving. But replication alone is not enough. Life also requires energy. In metabolism-first theories, the earliest systems were not genetic, but chemical networks. At deep-sea hydrothermal vents, minerals like iron sulfide can catalyze reactions between simple gases. These reactions can produce organic molecules and even primitive metabolic cycles. In this view, life began as a self-sustaining chemical system, with genetics emerging later. Another critical step is containment. Life requires boundaries. Certain molecules, lipids, naturally form spherical structures in water. These are called vesicles. They resemble primitive cell membranes. Inside these compartments, molecules could be protected, concentrated, and allowed to interact more efficiently. This may have been the origin of the first protocells. There is also the possibility that life began not in water, but on mineral surfaces. Certain clays, such as montmorillonite, can catalyze the formation of complex organic molecules. They can also organize these molecules into structured patterns. In this scenario, clay acts as a scaffold, guiding the assembly of increasingly complex systems. Each of these models addresses part of the problem. None fully explains it, but together they suggest a key idea. Life was not a single event. It was a gradual transition from chemistry to organized, evolving systems. If life required the right chemistry, then location mattered. Where on early Earth could these processes occur? Deep beneath the ocean surface, hydrothermal vents release mineral-rich fluids heated by Earth's interior. These environments provide chemical gradients, catalytic surfaces, and continuous energy. Here, organic molecules could form and accumulate in microscopic mineral pores. Some researchers propose that life began in these dark, high-pressure systems. On land, geothermal pools offered a different advantage. Cycles of wetting and drying could concentrate molecules. As water evaporates, chemicals are forced together, encouraging reactions that would not occur in dilute solutions. Ultraviolet radiation from the sun may have further driven molecular synthesis. Another candidate? Shallow lakes rich in dissolved minerals. These environments may have solved a critical problem: phosphate availability. Phosphate is essential for DNA, RNA, and cellular energy. In most environments, it is scarce, but in certain early lakes, conditions may have allowed phosphate to accumulate, enabling key biochemical reactions. Across these environments, a pattern emerges: energy sources, chemical diversity, and natural concentration mechanisms. Together, they created pockets of possibility, places where molecules could organize, interact, and evolve. By this point, Earth is no longer just a planet of chemistry. It is becoming a planet of systems, self-organizing, self-replicating, and increasingly complex. But something is still missing. We have the ingredients, we have the environment. What we do not yet fully understand is the exact moment when the first true cells emerged and left behind the earliest traces of life. That evidence comes next.
At this moment, something fundamental changes. Chemistry is no longer just reacting. It begins to organize. In the early oceans, simple molecules assemble into structures with boundaries. Lipids, naturally occurring compounds, form spherical membranes. These are not alive, but they are something new. They create separation, an inside and an outside. This process is known as compartmentalization. Inside these microscopic compartments, molecules are no longer dispersed in a vast ocean. They are concentrated, confined, forced to interact. Reactions become more efficient, sequences become possible. And now, two critical systems begin to converge: replication and metabolism. Replication allows information to persist. Metabolism allows systems to sustain themselves. Individually, neither is life. Together, they approach it. These early structures, protocells, are unstable, constantly forming and breaking apart. But over time, some combinations prove more resilient. Membranes become more selective. Chemical networks become more organized. Information systems become more reliable. At some point, there's no clear boundary. We cross a threshold from non-living chemistry to the first true cells. This transition is not recorded in fossils, but its consequences define everything that follows.
Once cells exist, evolution accelerates. The earliest life forms are simple, microscopic, single-celled organisms. They belong to two fundamental domains: bacteria and archaea. For hundreds of millions of years, these organisms dominate the planet. There are no plants, no animals, no visible ecosystems. And yet, life is already interacting, competing, and evolving. In shallow waters, microbial organisms form layered communities known as microbial mats. Each layer hosts different species. At the surface, some organisms interact with sunlight. Below, others rely on chemical gradients, consuming the byproducts of those above. This creates a vertical ecosystem, measured not in meters, but in millimeters. Within these mats, life becomes interconnected. Waste from one organism becomes energy for another. Over time, these communities leave behind physical structures. Layer upon layer of sediment is trapped and stabilized. Slowly, these formations grow upward. They are known as stromatolites. Some of the oldest evidence of life on Earth comes from these structures, dating back at least 3.5 to 3.7 billion years. They are not just fossils, they are records of ecosystems, of cooperation, competition, and chemical interaction. Life is no longer isolated, it is ecological. And then, one of the most transformative innovations in the history of life emerges. Some microorganisms develop the ability to capture sunlight. This process is called photosynthesis. Among them are a group known as cyanobacteria.
They use light energy to convert carbon dioxide and water into organic molecules. But in doing so, they produce something else, a waste product, oxygen. At first, this oxygen does not accumulate. It reacts immediately with dissolved iron and other elements in the oceans. But cyanobacteria continue to produce it relentlessly. Over millions of years, the balance begins to shift. Oxygen starts to build up. This changes everything. Before this moment, life is almost entirely anaerobic, adapted to environments without oxygen. Oxygen is toxic to these organisms. It damages their internal chemistry. But for others, it presents an opportunity. Oxygen allows for far more efficient energy production. A new metabolic pathway becomes possible, aerobic respiration. Life is no longer limited to low-energy chemical systems. It gains access to vastly greater energy. And with energy comes complexity. At around 2.4 billion years ago, oxygen reaches a critical threshold. It begins to accumulate in the atmosphere. This is the Great Oxygenation Event. From a planetary perspective, it is a transformation. From a biological perspective, it is a crisis. For many early organisms, oxygen is lethal. Entire populations collapse. Ecosystems are disrupted. This may be the first global environmental catastrophe caused by life itself. But extinction is only part of the story, because oxygen does not just destroy, it enables. It forms the ozone layer, protecting the surface from harmful ultraviolet radiation. It allows for new biochemical pathways, more efficient, more powerful. It creates conditions that favor new types of cells. And in this oxygen-rich world, a new form of life begins to emerge. Cells that are larger, more complex, and internally structured. These are the first eukaryotic cells, cells with nuclei and specialized components. Their origin likely involves symbiosis, one cell living inside another, forming a stable partnership. The rise of oxygen does not just change the environment, it reshapes the trajectory of evolution. At this point in Earth's history, something remarkable has happened. Life has moved beyond its origins, from simple chemistry to self-contained cells, from isolated reactions to interconnected ecosystems, and now toward complexity. Eukaryotic cells begin to diversify. They contain internal compartments, organelles, that perform specialized functions. They produce more energy, store more information, interact in new ways. This sets the stage for the next major transformation, the emergence of visible life. For billions of years, life has existed, unseen, microscopic, hidden within rocks and oceans. But soon, it will leave behind unmistakable traces, fossils, structures, evidence that can be studied, measured, and understood. In the next episode, we follow those traces to the oldest fossils ever found, and to the moment when life becomes not just detectable, but undeniable.